dobré ráno. It's the Czech good morning. Hopefully you enjoy staying in Prague and you had at least some time to see the city. And it's my pleasure today to introduce 5 speakers. 3 of them will be from the University of Minnesota. Then we will travel to France, to INRA, and then we will go back to US to the UC Davis. So first talk will be held by the Hayley Shanovich, and we will learn something about a filbert bud mite which destroys the hazelnut trees. Please.
All right, good morning, everyone. Thank you for that introduction. And thank you to the organizers. This is my first international conference, and I'm just excited to be here to talk to you about my research. So the United States is ranked 4th in the world for hazelnut production, with a crop value around 110 million euros, and that is mainly purchased by the snack food industry. Except 99% of the hazelnuts in the United States are produced in a single valley, the Willamette Valley of Oregon, Willamette Valley of Oregon, which is circled in blue on the map. They are one of the only places in the whole United States that is able to grow cultivars of European hazelnut, Corylus avellana. However, plant breeders in the eastern United States and Canada have been developing hybrids of the European hazelnut and endemic forest species of Corylus in hopes to combine the yield and nut quality characteristics of European hazelnut and the disease resistance and cold hardiness of the endemic forest Corylus species. So this has been underway for a while, and there are already cultivars of these hybrids that have been sold and are being grown throughout the Midwestern United States, which is filled in orange. So there are also processing facilities that have been already set up. And so there's kind of a burgeoning hazelnut industry already underway in the region. And so even though it's a novel crop in this area, so the hazelnuts have never been grown commercially here, growers are already experiencing arthropod pest pressure. And some of these pests are introduced species coming from the European hazelnuts from Europe, and some of them are naturally associated with the endemic Corylus species. Today, I'll be talking to you about one of the introduced species of concern, Phytopsis avellanae, the filbert bud mite, or big bud mite, as I'll be referring to it, which I have abbreviated BBM on my slides. So this is a areophid mite that forms galls of the vegetative and floral plant buds. So they feed on the inside of the buds and they turn them into galls through their feeding. And so in the picture on the left, you can see a normal uninfested bud, and then above it, 2 of the galls or big buds that they form. If you would take one of those and cut it open, you would see what's pictured in the bottom photo. You would see deformed necrotic tissue and thousands of these almost maggot-looking little mites running around. They're actually only 1/4 of a millimeter in size, so extremely tiny. You cannot see them with your naked eye. And so the above photo is a scanning electron microscope image of what those mites look like up close. There's also concern about these mites spreading into wild stands of our Corylus species in North America. We actually have found them in wild stands already, evidence of their galls. And there's concern that it could reduce the nut load of those crops and therefore decrease food available for wildlife, as hazelnut is an important food for wildlife in the eastern United States. So the life cycle of these mites is really interesting. They spend most of their time inside buds. So in the winter, they are overwintering and feeding and reproducing inside the buds. And then come spring, the nymphs will migrate from those galls into new axillary buds on the plant. And once inside new buds, the nymphs will molt to adults and then begin feeding and reproducing again. And so then by late summer, and especially in the autumn, after the leaves have fallen off the trees, you can already see the swelling of those new buds happening. So in this window, when the nymphs are migrating to new buds, there is this even smaller window of the peak mite emergence. And this is when the most of the mites are migrating to new axillary buds. And this is kind of the only effective time to use a chemical control for the mites. because otherwise they are safe inside of the buds. So growers are already experiencing problems with these mites in our region, and it's actually very hard for them to tell when this peak emergence is happening, as the mites are not visible with the naked eye. So we need to create a forecasting model or some way for them to predict when this mite emergence is happening so that they can target an effective spray. So my objectives were to characterize the mite phenology in the region. Hazelnuts have never been grown commercially anywhere else in the United States, so we don't know what the mite phenology will be like. Also creating a forecasting model to predict the peak mite emergence. So my research sites were experimental hazelnut plantings in the states of Minnesota, go figure, and Wisconsin. And so I had 2 sites around the star in southeastern Minnesota and 1 site in northern Wisconsin. Also, we didn't know when the mites would start exactly showing up. We knew from the literature they emerged in spring. And so we started monitoring in April, which can be a full-on snowstorm in Minnesota. And we sampled with double-sided sticky tape that we wrapped tightly around the branch above and below infested galls, like you can see in the middle photo. And we monitored basically throughout the season until the mites stopped showing up on the tapes. We also took plant phenology data weekly. So we measured the size of 5 randomly selected terminal buds on the plants weekly. And then once the season progressed, we counted the number of leaves that had leafed out on new shoots on the plant. We also recorded weather data via data loggers throughout the season. So when we would go out and change these tapes weekly, we would, when we take the tapes off, mount them onto slides and then count the mites under a dissecting scope. This project started right during the onset of the COVID-19 pandemic. So unfortunately, my kitchen table turned into my lab bench for about a year. It was actually difficult. So we had never seen the mites on the tapes before, and they're very tiny and kind of a translucent, maybe sometimes white pigmented color. And so as you can see in this photo on the right, just how small these are. And this is at 60 times magnification. And we had to play around with like different contrast of colors under the slides to see them better. And this is a very clean tape, but sometimes they're very full of debris like pollen and other things floating in the wind. So it wasn't as easy as we thought to count them. So to evaluate when the peak mite numbers occurred, we built models where our response variable was the number of big bud mites on those tapes. And then we tried different independent variables of time, like ordinal day, and then heat units, like accumulated growing degree days. For those calculations, we used a threshold temperature of 9 degrees Celsius, which was previously established by Weber and colleagues for the mite in 2008. And then since our sites are actually at 2 different climatic areas, we have southern Minnesota and northern Wisconsin, and they have different heat accumulation throughout the season. We wanted a way to standardize between those sites for the model. So we actually ended up using percent accumulated growing degree days. To do this, we used a local polynomial regression, or a LOES curve, as they're sometimes called. And so this is a way of fitting a smooth curve between 2 variables instead of trying to estimate exact parameters when you want to just estimate like a peak or maximum of a curve like we were trying to do. So my results are going to be presented as follows. So on the x-axis, I have the percent accumulated growing degree days, and on the y-axis is the square root of the average number of mites for each plant. So I would average the number of mites per plant per date, and then this is just the square root transform numbers. So in the blue panel are going to be graphs from northern Wisconsin, and in the yellow panel will be graphs from southern Minnesota sites. So in 2020, we found that at each site, the peak mite emergence was happening right around 40% accumulated growing degree days. We repeated the study this summer. And so this past summer, 2021, we found almost the exact same trend, that the peak mite emergence was happening right around 40% accumulated growing degree days. So this is cool. So in both years and across all sites, the peak number of mites occurred at the same percent accumulation of growing degree days. The consistency of that is really cool to me. And so this shows that this is a robust way to be able to predict the mites emergence across sites and years. So our next steps are to improve the model predictions by incorporating that plant phenology data into the model. And it will actually be really cool if those can predict peak mite emergence for growers to be able to use, because instead of them having to calculate accumulated growing degree days and then the percent of the growing degree days, they could just track the size of the buds or the number of leaves that have leafed out and use that as a proxy to determine when to spray for peak mite emergence. So in conclusion, the mites were successfully and uniformly predicted across sites using percent accumulated growing degree days, and we will see if plant phenology can also predict mite numbers. This will lead to a robust validated forecasting model for hazelnut growers to use throughout the Midwestern region. And so with that, I would like to thank all of my lab members for their amazing support. And I actually would like to highlight these three people who helped me so much with this project, counting so many tapes and so many mites. One of them is sitting right here, Patrick Parrish, and Phelan Anderson, who gave a talk on Tuesday. And yeah, I could not have done it without them. I'd also like to thank my funding bodies and collaborators in Wisconsin. And I'd be happy to take any questions or comments if there is time. Thank you.
Thank you, Haley. Any question, please?
Akif Eskalan from UC Davis. Very nice talk. Thank you very much. Our question is that, so apparently this is a new crop in your state. Is it replacing another crop, or it was like the empty land that they are planting there? Another one, is this mite problem in there where they are native to?
Yeah, both really good questions. So in regards to the, if it's replacing a crop or why they're being planted, I think in our region, in the Midwestern United States, it's honestly a lot of farmers who grow row crops, so corn and soybean, and a lot of growers are looking for a higher value specialty crop to start planting. And it's a very appealing crop for its environmental benefits to them. Our university, who's releasing a lot of these cultivars, markets it as being able to sequester carbon, be a perennial crop, and actually help a lot with runoff and water management in the landscape. And then as far as the mite in its native range, so it's native to most of Europe, wherever there are hazelnuts, basically throughout Europe and Western Asia. And so I actually haven't been able to find a lot of information on them in like the wild hazelnut stands, but they are a huge problem for the European hazelnut industry. And there's a lot of literature on them being problems in different countries throughout Europe and Turkey. Yeah, thank you.
Thank you. Some other question or question remote? Can I ask some, can I ask some students to volunteer to run with the microphone maybe? Is there someone who, yeah, thank you.
Yeah, thank you very much for this interesting talk. I am wondering if you find any inquilins or parasitoids or predators inside the gall, because we know that when mites are congregating, it is attracting predator and inquilins.
Yeah, that's a great question. So I have actually noticed, we have noticed as we've been counting the mites on the tapes, sometimes that there are predatory mites on the tapes. And a few times when I've dissected the galls, there are predatory mites inside of them too. I never see a lot of them, but I do see them, especially at certain time points throughout the season. I can't think of exactly when that was during our summer and spring, but I can't remember the name of the species, but yes, we have found predatory mites, and I'm not sure if those are native or maybe introduced as well. Yeah, thank you.
Thank you, Haley. We are running out of time, so thank you very much. And I would like to invite the next speaker from the University of Minnesota, Grace Hayes. In my program, no, it's not Grace now, it's August Kramer. Sorry guys, I misplaced you. Okay, and we will learn something about dispersal of bark beetle, mountain pine beetle in Montana, right?
Okay, thank you for that introduction. Greetings all, and thank you for joining me today on the last day of the conference. My name is August Kramer. I'm a master's student at the University of Minnesota, and I'm very excited to be here and share my early results from dispersal work on mountain pine beetle in the American West. So I'd like to start by introducing the organism. This is mountain pine beetle, Dendroctonus ponderosae. You'll see it abbreviated in my slides as MPB. It's a native insect to Western North America, and it's very aggressive there. It mass attacks most of the Western pine species we have. And at this point in the conference, we're all pretty familiar with bark beetles, so we know that they're feeding beneath the bark in the cambium layer. We know a little bit about long distance dispersal, but not very much. Flight mill studies have shown that mountain pine beetle can disperse through powered flight about 24 kilometers, but we really don't know anything about long distance capabilities beyond the fact that we think that it's probably operating in high elevation wind currents and passively dispersing very long distances. Mountain pine beetle also has what's called an eruptive population dynamic, which means that it exists at very low levels for extended periods of time in the landscape. And then when conditions are right, it will experience a population boom and cause many, many millions of hectares of damage. And so on the right of this slide, you can see the insect, and on the bottom right, you can see what's called a pitch tube. And that's the entry point of the beetle into the tree. And the tree is actively trying to eject the beetle as it's boring in. And this is what the damage can look like. This is a site in southwestern Montana. And all of the gray that you see there is mountain pine beetle killed pine. You can see that there's not very much left. But I'm focused on dispersal. And so this figure might look familiar. Brian Okama presented this on Tuesday. And as he explained, the traditional range of mountain pine beetle in red here is been relegated to the Western United States and Western pines. With climate change, we're seeing movement into more northern latitudes and also into higher elevations. And that's indicated by the blue polygon to the north. I'm focused more so on the red arrow in the middle of this map, which is the dispersal of mountain pine beetle across the Great Plains of the North American continent, which is symbolized in this map in yellow. And this is a photo of kind of the interface between Western forests and Great Plains. You can see that there are pine trees that are extending out into the plains for a short while, and then it gives way to several hundred kilometers of just grassland that's punctuated every once in a while by a sporadic pine forest or a city or a town that might have some pine planted as well. But my questions for the study are three. The first is, can we catch mountain pine beetle from an active infestation? The second, how far from an infestation are they flying to arrive at a trap? And third, do distance and forest cover predict the number of beetles that I can find in a trap? And so I want to talk about my study area a little bit. This is the United States, and I did my study in Montana, which is located here. Within Montana, I focused on a portion of the state that was kind of north central, northwest. And we did this for a couple of reasons. For one, we knew that there was mountain pine beetle active in this area, but it also allowed us to test a variety of forest conditions. So in the western half of this transect, we had some pretty typical western pine forests. But as we move east, and especially the three easternmost sites, those being Augusta, Fort Shaw, and Sun River, you essentially get out into that grassland, that Great Plains ecosystem. My trapping methods were as follows. I set 20 Lindgren funnel traps, approximately 24 kilometers apart along a 290 kilometer transect. Each one was baited with a standard mountain pine beetle lure. And I set those traps at the end of July and checked them weekly until the end of August, which coincides with the peak flight period for mountain pine beetle. And on the right here, you can see a variety of my trapping locations in pine forests in Montana. And we don't have to focus too much on the specifics of this table. I just want to illustrate a couple things. The first is that I did catch mountain pine beetle. I caught about 3,700. So the first question can be answered affirmatively. Yes, we can catch mountain pine beetle, which is great because that paves the way for my second two questions. The other thing I want to say is that when we arrange these trap sites from west to east, we can see that the majority of the beetles are occurring in that western half of the transect. Not very many turned up in eastern forests, which is more or less what we expected. But again, with this positive answer to question 1, we can move into questions 2 and 3. And I addressed those using a little bit of GIS. The first was to measure the distance between a trapping site and the nearest mountain pine beetle infestation. And I did that using aerial detection survey data, which is provided by the US Forest Service. And what that data is basically is just an aerial footprint of the previous year's damage or mortality. And so on this map, you can see the red polygons are mountain pine beetle. And it's a pretty simple operation to just measure the distance between those polygons and a trapping site. The next thing I wanted to do was look at the percent forest cover and percent host cover within 1.6 kilometer and 3.2 kilometer radii of the trapping sites. You can see those concentric circles representing those radii. And within those, there's kind of a mosaic of green polygons, and those represent different tree species that are present on the landscape. As it turns out, bark beetle dispersal is a really complicated phenomenon, and 1 or 2 variables is not going to really describe exactly what's going on. As it turns out, actually, most of the variables that I tested ended up not being significant, and that included distance to the nearest infestation of any size, percent forest or host cover within any of the tested radii. And what I ended up doing instead was to take a broader view and look at some of the more general trends. And what we found was that the majority of our beetles were captured at trapping sites where forested area within the 3.2 kilometer circle was greater than 60%. And so on this graph, on the x-axis, we have the condition of less than 60% or greater than 60% forested. And on the y, we have our beetle catch. And the other thing that we did was we found that beetle catch increased if the nearest infestation was within 10 kilometers of an active infestation. Again, on the x-axis, we have less than 10 kilometers or greater than 10 kilometers, and our average beetle catch on the y-axis. You'll notice that our adjusted R-square values were fairly low for this. And what that just means is that there's a bit of variability in the data, and that these findings are only explaining a portion of that variability. Now, there's a couple ways that we can look at this. One of the ways, I think, is to look at the big bar, basically. And we can say, okay, a majority of the beetles are behaving in a certain way. They're mostly staying close to their natal host, and they're mostly infesting or trying to infest areas that have a lot of trees. I think, more interestingly, we can look at the small bar. We can examine the minority group that is flying very long distances and is arriving at locations that really don't have any trees at all. And there are a couple of sites and a couple of beetles that I'd like to talk about. So I want to revisit the transect just so that we can place ourselves geographically. The 2 sites that I want to talk about are Augusta and Sun River, and those are located here, out in the Great Plains. At Augusta, we caught 1 mountain pine beetle during the first collection cycle. And this is what that site looked like. Again, it was essentially out in the Great Plains. It was 53 kilometers from the nearest putative source of mountain pine beetle. And it's not hanging in a pine tree. That's a willow, that's a salix. It's not where we would have expected to find mountain pine beetle at all. And so we know that this beetle is a female, and it's very interesting that it arrived at this trap. We can presume that it was a long distance disperser looking for a host and potentially arrived there on a high elevation air current. The other site I want to talk about is Sun River. Now, Sun River is interesting for a couple of reasons. And I'll start by just saying that my broader research includes another species of bark beetle that I have not mentioned yet. And that's Ips grandicollis. And Ips grandicollis is an eastern species of bark beetle that has been slowly advancing over the last couple of decades into Western forests. It's interesting because this trap that I'm showing you here was part of that research on Ips grandicollis. At each site, I set additional traps baited with an Ips grandicollis lure, and the mountain pine beetle that I caught at Sun River was actually in an Ips grandicollis trap. So there's a small body of research that examines how mountain pine beetle is behaving at very low population densities. And one of the things that we think might be happening is mountain pine beetle will respond to other species' pheromones and take advantage of the strength in their numbers when they don't have enough to overcome the defensive capabilities of a host tree. And so it's interesting that this one beetle that we found in the wrong trap was very, very distant from any other mountain pine beetle population and was very distant from any other host material as well. This trap was 47 kilometers away from the nearest population of mountain pine beetle. And so what we can say from this is that the rate of long distance dispersal is likely very low. We only had 2 that were roughly 50 kilometers away from a source out of the 3,700 that we caught. So the vast majority of the beetles that we catch are staying within 10 kilometers of their natal host. And that's very significant if we're thinking about risk assessment, because we can say that the risk of new and very distant infestations is likely really low. Beetles tend to stay very close to their outbreaks, but the fact that we had a couple of beetles that were responding to multiple pheromone cues after presumed long distance dispersal would suggest that there is not only a possibility of establishment in new forests after long distance dispersal, but that it could even be facilitated by eastern species that are operating in their own range. And there's a lot more to do. I'm very excited to begin the next steps in my analysis, which include looking at the metabolic budget of individual beetles. So I'll be examining what kinds of powered flight metabolites, things like carbohydrates, proteins, and lipids exist within those insects that I've captured. And I'll also be examining some of the similarities and differences between eastern forests, western forests, and central forests and the insect communities there to see what kinds of friends or foes mountain pine beetle might encounter on a hypothetical journey across the Great Plains. And with that, I'd like to thank all of my funding sources. I'd like to thank the logistical help I got from state and federal agencies in America, the fabulous lab group at the University of Minnesota, and the organizers for putting on this great meeting. Thank you.
Thank you, August. Questions here?
Thank you for this really exciting talk. I have one question and perhaps one remark. The question is about the relationship between the forested area close to the traps and the amount of beetle caught. Is it any type of forest or is it a conifer forest?
Yeah, so Montana is, as far as American forests are concerned, Montana is not terribly diverse. Usually it is some mix of either ponderosa pine and lodgepole pine, which are host species, or Douglas fir, which is a non-host species. So the forested area includes any type of forest cover. And then the host cover was an attempt to kind of separate those out and look just at available host material for mountain pine beetle.
So, but basically there were conifers there.
Yes,
Okay.
yes, that's correct.
And then perhaps a remark. I have done transects myself too, and what I observed was a high variability in the catches of traps positioned close to each other. And I was wondering if you increase the number of traps in each of the locations of your transect. I was wondering whether you might be more successful in catching more insects. There seems to be a small scale variability in the catches.
That's a really good point. And there was an awful lot of variability within the transect. I would like to trap more if I can. And in fact, just last summer, a month ago, I was back out in Montana and I extended my trapping effort into further east and then also into a new area a little further south. But I like the idea of setting more traps at the same locations to see if you get consistency there. I think that'd be really interesting. One thing that I want to be careful of is spillover, because mountain pine beetle is so aggressive. Drawing them into a forested stand can sometimes lead to, unintended tree mortality. And when I'm working on public land, I find that the landowners or the land managers don't tend to invite me back when I kill their trees.
Question, do you have any idea of the rate you found in 10-kilometer distance from the beetles, from the number of beetles you caught?
The proportion of beetles that were caught that originated presumably from within 10 kilometers. Is that the question?
Yeah.
Yeah, so it's almost all of them. 99.99% of beetles were within 10 kilometers. Only 2 were beyond that.
But this is an estimation, or do you use recapture studies?
So I think these data that I was presenting, it's a sample. And so it's what I've been able to observe and find. I'd be curious to know if there are other examples of this in the literature and to see if people are catching similar frequencies of, or similar, I guess, proportions of beetles as close as I am. But I think from what I know, this is what I've been seeing.
And did you, my last question, did you record any abiotic things which could influence the flight direction?
That's a really good question. There's some pretty interesting research looking at like kind of landscape geography and topography, wind direction, things like that, and how all of that influences mountain pine beetle dispersal. I have not addressed that with my study, but it's definitely something that I'm interested in and have been reading about. And I think is very, very relevant to mountain pine beetle dispersal. Thank you.
Thank you. Okay.
Oh, yeah.
There's a question from Etsuko. Is there a sex ratio bias in the response to pheromones?
That's a good question. I don't know for sure. I know that in the beetles that I captured, the sex ratio is roughly 1-to-1. They're pretty much even. But I'm not sure if that is consistent across other studies. Okay, thank you.
And now I would like to invite Grace. And we will learn something about invasive exotic Longhorn borer, Tricho Ferrous Campestris in Minnesota. So,
thank you so much for that introduction. I'm Grace Haynes from the University of Minnesota. I promise I'm the last University of Minnesota representative. And I'll be talking to you today about Tricho Ferrous Campestris, an invasive or exotic longhorn beetle. I was going to start my talk by introducing many trade, the way that the international trade can facilitate invasive species movement, but I think that that's been pretty well covered in the rest of this conference. So I'll assume that everyone here is familiar with the fact that international trade, as pictured in this map, can facilitate invasive species. The invasive species that I'll be talking about is Tricho Ferrous Campestris. Its common name is velvet longhorn beetle, and I'll be referring to it as VLB, its acronym. And this is one such beetle that can be introduced via international trade. It likely travels via pallets and solid wood packing material, and it comes from Asia and Eastern Europe, parts of Eastern Europe, and it has gradually spread west over the past few decades. Its native range is a little bit unclear from the literature, and so that's why I can't go into more detail about specifically which countries it's native to versus where it's invaded. But in general, the trend is that it's moving westward. And it's unknown what its impact can be in its invaded range, and that's reflected in its variable regulatory status in different parts of the world. So in Europe, where it's invasive, it's still regulated as a quarantine pest in all of the countries on the right side of the screen that are highlighted in green. Whereas in North America, you see kind of different trends. It's unregulated in the United States, with the exception of Hawaii and US territories because of how separate they are from the rest of the United States. But in Canada, it's still considered to be a quarantine pest. And one way that it is able to establish in so many varied environments is because it has such an extensive host list. So it can infest, the literature states that it can, excuse me, can infest over 40 genera of trees. And that actually includes Robinia pseudoacacia that we've been talking about this week a lot, as well as everything pictured here. And it reportedly favors mulberry and apple trees. But the thing is that much of the literature that discusses these preferences is observational studies and descriptive studies rather than controlled laboratory studies. And so that leaves a lot of room for us to come in and investigate in a laboratory environment. So our research has focused on two primary categories: phenology in the novel range, as well as host preferences. We don't have time to get into all the results that we've found, so I'll just stick to the most relevant findings. We started both components of our studies by collecting live adults in Lindgren funnel traps baited with pheromone. And first we used that capture timing to describe phenology. So similar to the modeling that Haley was talking about, we calculated degree days and accumulated heat, and we based that on a 10 degrees Celsius developmental threshold, which is typical of Longhorn beetles. These are the results that we found. So on the left, you see 2019, on the right, you see 2020. And then on our x-axis, we have the accumulated degree days. And on the y-axis, we have the average number of VLB that were caught weekly. And so the main points that I want to draw your eye to in these graphs are that we have a single peak both years, here and here, and that peak occurs at approximately the same number of accumulated degree days. And so this can be really helpful with monitoring. So as this beetle likely continues to expand its range to additional locations, and we may be trying to monitor for it, see how far it's spread, it's important to know when it's peaking so that we can adequately catch it and put our resources to their best use. Looking at our host research, that can be divided into two different components as well. So we have our choice studies and our no choice studies. The choice studies consisted mainly of looking at where VLB chose to lay its eggs among the tree species that we tested. And the no choice consisted primarily of seeing how many VLB emerged from each of these same 4 tree species when they were placed on it without being given a choice. So I'll start with our choice study. First of all, these are the 4 tree species that we chose. All 4 of these genera were reported in the literature as potential hosts. And so we chose black walnut, Juglans nigra, because it's likely been a component facilitating the spread of VLB in certain cases internationally. Honeycrisp apple, or Malus domestica, is an important economic species in the upper Midwest where our study was located. And then Acer saccharum and Pinus strobus are both very important ecologically in that same region. So that's why we included these 4 tree species specifically. So going back to our choice study, our ovipositional choice study, we used those same VLB that we caught in those live Lindgren funnel traps baited with pheromone. We mated them with each other, and then we placed these mated females in the center of a Tupperware container, as pictured, with these approximately 10 centimeter long segments of each of these 4 tree species. And then we allowed the female VLB to oviposit on all of them until she finished ovipositing, and then we counted our eggs. So here we have our results from our choice studies with the number of eggs laid on the y-axis, and on the x-axis we have these 4 tree species that we tested. And you'll see that we had significant results both years, and that they're relatively consistent as well. So both years we saw significantly more eggs laid on Juglans nigra than Pinus strobus. And numerically, we saw the largest number of eggs laid on Euglens nigra both years, although our statistical differences have some changes year by year. But importantly, Euglens nigra had the most eggs laid both years. Then looking at our no choice study, the way we set it up was we placed 3 eggs each on replicates of these same 4 tree species. So that's pictured in this diagram on the bottom. And then we took various measurements of larval performance over the course of their development. So on the top left, you see we peeled our logs, and those are some galleries. You can really see the way that they just destroy this wood. They completely destroy the wood, and there's galleries everywhere. In the center, then, that's a larva that we took out of a subset of our samples. We measured larval length and some other components of their well-being. And then on the top right, you see them emerging from one of these logs. And that is what I'll focus on, those results today. So we saw, here are our results. Once again, we have 2019 on the left, 2020 on the right. We have our host species on the x-axis, and this time on the y-axis, we have the total emerged Trichoferous campestris, or VLB. Once again, we have significance from both years, and then we're seeing somewhat similar trends with a greater number of VLB emerging from Malus domestica and Euglens nigra than from Acer saccharum or Pinus strobus. And that's consistent between both years. But interestingly, in 2019, we saw more emergence from Malus domestica numerically than any of the other tree species represented. Whereas in 2020, you'll see that our highest number on average was from Euglens nigra instead. So to summarize, when looking at phenology, we observed maximum catch occurring in early to mid-July, and that's at approximately 650 degree days. And so when sampling for this in a potentially novel location, if that is similar climatically to Minnesota, that might be a good approximate time to try and set traps out. And then when looking at host studies, interestingly, we saw emergence and eggs laid on all 4 tree species. So theoretically, any of these tree species could be hosts. But it does seem that Malus domestica and Euglens nigra may be slightly more suitable as hosts than either of our other species, Acer saccharum or Pinus strobus. So with that, I'd like to thank my lab and my funding sources, as well as the conference organizers. This has been a really amazing experience. And I'll take any questions if there's time. Wow. That's really interesting. All right.
Maybe I would have one question as well. In the trap catches, did you use a specific pheromone, or did you use this generic mixture for Longhorn beetle?
So the pheromone that we used was developed by a team led by Annie Ray. And I believe that it was specific to Trichoferous campestris, although I don't think that we have a lot of similar beetles that would be likely to be attracted to that in the United States.
Okay, thank you very much, Grace, for the nice talk. And I am happy to introduce Alan Rock, and we will continue in Longhorn beetles, right? And we will learn about using this generic mixture or generic pheromone for monitoring of longhorn beetles in Europe, right?
Thank you for the introduction. So I will continue, and you will see that we have a great potential to trap longhorn beetle all over the world. It is my talk, but in fact, there are 47 co-authors. You will have the list at the end, because it corresponds to a joint effort to trap worldwide. And here you have some species that have been trapped and they are invasive in a continent or another one. So I think there is no need to comment this slide, because everybody here is aware of the increase, either exponential in Europe or linear in some other countries. What is interesting is that we added China recently to this graph, and in China it's the same process, even if it is at lower with a lower amount of insects. But anyway, it's the same process of increase. And this, despite of strong differences in border controls. For instance, Europe is not looking at species that are not on the quarantine lists, but it is not the same in New Zealand, for instance, where everything is controlled. But maybe the most important, and Jiri yesterday told already about this, the most important is to consider that now a lot of species that are arriving are what we call emerging invaders, because these species correspond to new routes of trade, either new plants that are traded or new countries that are included in global trade. And so these species are rather difficult to be spotted. If you use, for instance, specific pheromones, you will never get them. But the problem is that they are arriving. Most of them, Anoplophora glabripenis, for instance, the Asian longhorn beetle, before it arrived in the US, was never spotted anywhere in the world, except in China, its native range. And so a lot of species are like that. And we are facing a big challenge to be capable of detecting these species at arrival. And they cannot be expected from the list of interception, most of them. A lot of species, I can say even more than 50% of the species that established during the last 20 years were not included in the list used for interception, especially in Europe. And so the trapping, I already said, cannot rely on specific lures, and we need new strategy. One of this strategy, it's not the single one, we have also sentinel plantings and others, it's automatic trapping at borders using lure, but lures with a generic attractiveness, if we can, for xylophagus groups. And there is one group which is very well adapted, it's Cerambycidae. Why? Because recently there have been very large progress, especially by Larry Youngs and Jocelyn Miller team, and Annie Ray, she just cited before, about the identification of pheromone of Cerambycid, both sex and sex aggregation pheromone that can attract either male or female or both of them. And what was interesting is that Jocelyn and Larry demonstrated that a lot of these pheromones are well conserved at world level. It means that you can trap species in China using pheromone of a species in US or Europe, given it is the same genus or the same tribe. And here you have some compounds that are interesting from this point of view. I did not detail, but you will see on the PPT when it will be released. There are 8 compounds that we used and we tested in a first experiment in France during 2013 to 2018. with increasing combination, 1, 2, 4, 8 compounds. And finally, we found that there are no repulsive effect when you are combining the 8 pheromones together. So if there is no repellency between components, we can use them as a blend with a generic attractiveness. And you can see here the generic attractiveness. It was an experiment in France. We trapped more than 50% of a Longhorn beetle native to France. But more important, most of the tribes were well represented. We caught all the species in some ceramicid tribes at 100%, 10 tribes, and more than 50%, 15 tribes. At the same time, we trapped 8 exotic species. You can see, for instance, for Clitini, which is an important tribe here. This is the invasive Xylotrichus tebingi. We trap quite all the species, and there are a number in France. There are 22 species, and we trap 18 of these 22 species. So after that, the idea was to extend this. And hopefully, we were lucky. We had 3 successive European projects that allowed to develop a worldwide trapping program. And so at first, we had a trapping program in Europe at the ports of entry. Then we used the multilure as a control for another project about ambrosia beetles in the Mediterranean. And finally, we had the OMET project led by Hervé, who allowed us to generalize the trapping at world level through partners abroad Europe. And the rationale was, if a species is regularly trapped in numbers by the blend on a continent, higher is the probability to detect it at arrival on other continents. And if the blend effectively shows a generic attractiveness, unknown species could be trapped. So the trapping network is this one. It's a rough design, but anyway, we cover quite all of Europe, quite all of China, 2 states of US, 1 part of Canada, Siberia, Australia, and part of the Caribbean. And we use everywhere the same design with 1 milliliter of blend on a dental cotton stick, and the wool being into a polyethylene mini zip bag that allow the volatiles to go away, and a black Lindgren funnel trap. And the results, so far we trapped 335 ceramicid species without the results from this year. And we capture them from the most important beetle subfamily with an effective genericity, as you can see. And the more one, we were Clitini, Leptorini, but also the interest is Monocamini here, because you can see in the next slide that we trapped most of the species that are interesting to survey the expansion of the pinewood nematode. And more than 100 of the species were trapped with more than 20 specimens. So we can say that the probability to trap them when they arrive is high. Some example of important genera trapped worldwide, 11 species of Monocamus. Most of species that are considered as the one that are spreading the pinewood nematode. So in China, in Northern America, in Europe, here in Russia, 13 species of Grylotrichus, which are also important invader, 11 species of Phimatodes, and interestingly, we trap species both in the native range and in the invaded range, meaning that when we say the rationale is if we trap them in the native range, we can trap them in the invasion range. It is true. And for instance, this one on the left, the Xylotrichus chinensis is a major pest of Morus. We trap it in Beijing, but also in France, in Spain and Greece. And the other one, which is a European species widespread all over Europe, we trapped it in Canada, in Ohio, and in Michigan with the multi-layer blend. So we think now that such a blend is a good tool for early detection of non-native for some tribes. But these tribes are maybe the most important for the forest protection because they include Clitini, Calidini, Monocamini, Acanthocini, Acanthodirini, and Prionini. But we are testing the addition of new pheromones inside the blend. For instance, the pheromone of Aromia bungi, trichopherone for Trichoferus, and I told you just before that we succeeded in trapping all the Trichoferus species in Europe to improve the detection scope. But some tribes are not trapped at all with the lure. And we have also to take this into account. Obviously, Apterus species are not trapped because they cannot fly to our trap. But we have also some other species, mostly related to herbaceous plant rather than trees. And so that it's interesting to be considered. But also some other species need specific compound, highly specific compound. For instance, Rosalia, which is protected in Europe, has a special pheromone. We tested it and it is working. And we tested it adding to the blend. It is also working. It cannot decrease. It did not decrease the other species, the trapping of the other species, but we trap at the same time Rosalia and we let them go without any problem. But at the end, you need to keep in mind that traps are only an additional tool because the probability that an insect is arriving at the right stage to be trapped in the ports of entry is limited. And so what we are suggesting is to settle the traps in the garbage places in the ports or near the ports in order to allow the larvae to complete their development. And so they turn to adult and they can be trapped. And it may increase the trapping detection success. So the contributor, there are 2 slides because there are many, many, many. First from abroad Europe, China, Russia, Michigan, Ohio, Nova Scotia, Australia, Martinique. But also from Europe, Austria, Bulgaria, England, France, Greece, Italy, the Netherlands, Norway, Portugal, Slovenia, Spain, Sweden, and Switzerland. And finally, I have to thank Jocelyn Miller and Larry Youngs because it was from a discussion with them during a USDA meeting that we decided to do this. And I think it's very interesting for all of us. So thank you for your attention.
Thank you very much, Alan. Questions? Yes. Here, please, Jean-Claude.
Yeah.
Excuse me, the mic is not working.
Yeah. In fact, what I presented here is the wool design. I mean that traps were, for instance, for France, first put into the forests because we consider that the probability to get invasive species inside a port is completely random. we cannot say it will arrive. And so if you want to get statistical treatment, you need to put it inside the forest. But what we did for the ports is that we put traps inside the port and trap outside the port in the first place where we can find either broad-leaved or conifer, not stand, but wooden area. And so it's replying to your question. With a condition is that we need to analyze the genetics of species we are trapping inside the pot and the ones that we are trapping outside, because some species can come from outside to the pot, and so and obviously the converse. So we are doing what you say, because effectively the maturation feeding is important, and what I can say also is that in the ports we trapped already a lot of invasive species with this multilure, but in the garbage area.
Thank you, Alan. Some more questions? I maybe would have one. It's the first time when I have heard about one universal, like generic pheromone mixture. Coming back to the biology of these species, didn't they show the cross attraction to each other's pheromone?
Yeah, you are right, but it's the principle of multilure blend. We are interested in this. The only repellents, it would be important to check if there is any repellency, and we did this. And finally, we found that the only repellency we found was between ethanol, because we added, I didn't say this, but in a number of cases, we added ethanol and alpha-pinene in order to trap at the same time bark beetles. But for some species, such as Phimatodes testaceus, we found repellency of ethanol, not of the combination of the pheromones. And another point is that we expected to do the same with buprestids, but it is not working. There is no generic combination. And for bark beetles, it's a bit more complicated, because you have to include the volatile from the trees and so on. But we expect for bark and ambrosia beetles to go in this direction.
I meant, what happens if these species will meet in the nature? Will they attract to each other?
Yeah. I think for Phimatodes, it would be interesting in the US to look if there are hybrids with the native species. But for the moment, I don't know. We only trap them. And for Tricho ferrous, it will be the case also. If Tricho ferrous Campestris is effectively established all over Europe, there are, for instance, in France, there are 5 other species of Tricho ferrous, and it would be good to look for the possibility of hybridization.
Thank you for answer. Some more? So I would like to now invite the last speaker of this session. Thank you,
Excuse
Alan,
me,
once more. I would like to introduce Shannon Lynch from the UC Davis in California. And she will speak about phylogenetics and epidemiology tools which can help to predict the pest spreading, right?
Yes. Thank you. Thank you for that introduction. And thank you to the organizers for your persistence in making this event happen. My name is Shannon Lynch. I am a postdoc at UC Davis in the plant pathology department. And I am here to talk about how we took an evolutionary ecology approach to try and predict the establishment of a multi-host pest pathogen invasion in Southern California, and to try and potentially that could be used as ways to improve decision-making in invasive species management more broadly. So the complex is called Fusarium dieback. It is caused by the combined effects of these specific Fusarium species. that are vectored by these Eualacea ambrosia beetle species, the Polyphagus shot-hole borer and the Kuroshio shot-hole borer. Together, they're commonly referred to as the invasive shot-hole borers in California. The beetles pack the fungal spores into the special mouth parts called mycangia, where they carry the spores from tree to tree to grow their food in their natal galleries. The beetles were introduced to California from Southeast Asia in 2012 and 14, presumably on packing material. I don't think this was the direct route that they took, but that's where they landed. And the impacts of these of this complex can be quite devastating. So I'm just going to show you the worst case scenario here. This riparian forest in Southern California. Within 1 year after attack, the beetles killed over 200,000 trees. And we see cascading effects of this mortality as well. So this is standing on the ground at that same site, where 9 months later, we see this encroachment of castor bean, which is a highly invasive weedy plant species that prevents recovery and recruitment of the native willow species, which are serve as critical breeding habitat for endangered bird species. So the complex has a very broad host range. I just put up a collection of host species up here. This isn't everything, but just to show you kind of all of the different symptoms that can be produced depending on the host. So broad host range, 77 species, tree species support beetle reproduction, or what we refer to as competent hosts. And 20 of those species are native to California, and they dominate the landscape. Another important host is avocado, because California produces 90% of the domestic crop in the United States. And then roughly between 25 and 60% of all of the individuals that are planted in the urban forests are competent hosts as well in California. So we know that these host species are important epidemiologically, but they also have a phylogenetic structure from our previous work. So I'm just going to present to you kind of the overall principle here. If you want to know more detail about it, you can look at our most recent paper in Evolutionary Applications. But in general, the idea is that more closely related species are more likely to share similar pathogens because they are more similar in their traits. So for example, this group in the orange and in the blue, they are more likely to share similar pathogens because the phylogenetic distances between them are shorter. But the blue group and the orange group are less likely to share pathogens between them because the phylogenetic distances are longer. And so what this phylogenetic piece is doing is it's explaining the relationships, and as a first approximation, it can tell us which species are going to be impacted and to what extent. And because the Fusarium dieback hosts are distributed across this complex landscape, where in some areas you might just have one species that, like this avocado grove, or you might have a lot of species like this native riparian habitat. Our aim was basically to see if we could use this evolutionary understanding of host range to try and predict where the complex is most likely to establish and have the greatest impact. And there's good reason to do this. So this is a distribution map in California, just showing you where we are for reference in that blue box on the lower left corner. And recently, about 250 kilometers north of the leading edge of the infestation, we detected some beetles in traps, most likely through human-mediated dispersal. They haven't yet been found on any tree species yet, but that's why we're doing this work. So to do this, we established a very large network of permanent monitoring plots all the way from in Southern California, from the border, Mexican border in San Diego County to the north in Ventura County. Up here, the plots are in infested and non-infested sites in avocado groves and also peri-urban and wildland forests to basically capture the entire environmental range for which the beetles and fungi could thrive. We also are measuring microclimate in each of those plots, and on every tree, we're measuring a bunch of stuff, but in addition to all of that, the amount of disease of present on each tree in the plot. And then we basically are estimated the site susceptibility based on these interaction between phylogenetic structure of a local community and host abundance. And so if you look at this, imagine this is like a local site. These are the species that could be found at that local site. And then basically what we did was we calculated the probability that each species at that in that local community is a host. And we did that by, this Death Star looking thing is a phylogenetic tree of all of the woody shrub and tree species for which the beetles can encounter in California. We calculated the phylogenetic distance between each of those focal species and all of the confirmed hosts that are affected. We weighted that by how abundant each species was in that local community, added those up, and that gave us an estimate of site susceptibility. And so when we did this logistic regression, looking at the probability that a site is infested, we have this predicted probability and our observed probability in infested and non-infested sites. And we have the susceptibility based on that phylogenetic species composition on the x-axis. And we have this highly significant effect in which we see an increasing probability that a site is infested with increasing phylogenetically weighted host abundance. Another way to look at this is histograms of all of our plots that are infested and non-infested plots here. And what we see is the skew where places that we expected to see disease, we actually see disease. And more of the non-infested locations land in those kind of lower estimates of phylogenetically weighted host abundance. So the community context and community composition alone does a pretty good job of explaining things. It's not perfect because at some of these sites, we don't expect to see disease and we do. And then in other sites, we expect to see disease and we don't. This could be an artifact, of course, of time. It's just not gotten there yet, but we also know that temperature has important constraints on beetle development as well. And because we measured the microclimate in each of our sites, we were able to estimate the number of generations that each site could support using a degree day model, as well as parameters that were experimentally determined in previous studies. And basically, we would expect that sites where you see the temperature landing in that kind of optimal range more often is going to support more beetles, in contrast to like this example here of a non-infested plot. So we added that to the model, as well as the interaction between community context and microclimate impact on the beetles. And in our logistic regression of infested and non-infested sites, we had a highly significant effect on all three. We did a kind of a preliminary sensitivity analysis, and it looks something like this. So this is a scenario where a site could produce one beetle generation, so the climate is not that favorable for beetle development. But if you have a site that can produce more beetle generations, we see this interesting effect where sites where we wouldn't expect to see disease being, a site being infested, just based on that community composition alone, there's an increase in the predicted probability that it's infested because the site is, the environment is more favorable for beetle development. And so we see this kind of really cool, interesting effect of climate depending on the community context in which these places that, warmer places that can produce more beetle generations are going to allow an infestation in sites that are otherwise unfavorable. So it's a really kind of an interesting interaction. This is just kind of give you a sense of like what our sites look like more closely, just to give you an example. And what we have here is the proportion of trees that are infested in each of these places with the probabilities that are on those sites. And so we see kind of in those higher probabilities, they are actually in reality infested, and those locations with lower probabilities are less infested. And so as far as like practical applications, like in a site like this, where we don't see anything really just yet, the idea is like, where can we prioritize surveillance efforts so that we are being most efficient and effective at detecting? And so, you know, for something like this, we could say, why don't we just really focus our monitoring efforts, like, you know, at these sites that have higher probabilities? That's the basic idea. So this phylogenetically informed predictive model, I think, can be a powerful tool to understand complex interactions, as well as an interesting way to assess risk and inform response priorities. So in California, it's being used to basically target different control measures and decide where those should be implemented across a landscape. Can decrease the use of systemic pesticides by targeting their use, and also prioritizing where costly and time-consuming tree removal measures would be the most strategized, where those would be best implemented. And then it can also potentially be used to try and predict where things, what's going to happen in other places around the world where the beetles have established. For example, the polyphagous shot-hole borer is in Israel as well as South Africa, and also can be used as a way to identify where what would happen if it were to get somewhere, for example, like Mexico, that has a lot of similar host species in California and also has a very large avocado industry. And so currently what I am working on right now is, because we want to know what's going to happen across the state of California. So I'm using this to make predictions across the state. We're also sampling these sites over time. So we're going to include that temporal piece. And then I'm also working on including landscape considerations into the model. So in addition to, you know, combination of disease and non-disease trees at a particular site, I'm working on developing a phylogenetically informed index of connectivity to look at the relative importance of landscape configuration in the establishment and spread of the complex. So I just want to first thank my field crew for this heroic effort in all of the adventures that we've had along the way in sampling these sites. This research is the outcome of a really huge multi-campus and multi-agency collaborative effort. So it's the outcome of a lot of interactions with stakeholders and land managers that has informed our approach to research and addressing this problem. Yeah. And I also just wanted to give a shout out to the bark beetle microbiome working group. And we have a meeting September 29th. So if you're interested in beetle fungal symbiosis, we're going to be talking about how science can help regulators. And so it's going to be an interesting conversation about applications of the science into policy. So definitely join us. And you can find more information on the website, and I'll leave this up and happy to take any questions. Unless you want coffee.
Yeah. Thank you, Shannon. Some question? In this group is also involved Jiří Hulcer, right?
Yes.
Yeah.
Yes, Jiří and our colleagues from South Africa are the PIs on the project. And Romina has a question.
Hi, great talk. I'm wondering if you are looking into urban forests. Like Florida has a very different forest composition than California, but our urban forests are very diverse and might overlap a lot in the species.
Yeah, thank you for that question. So yes, there's a couple ways we are looking at the urban forest. That's on another project completely, but we're using Forest Service urban forest plots to also incorporate some of these considerations into spread, because also the urban forest is important. It is like the matrix is conducive to spread, and that's going to be really important in understanding impacts in California for sure. Yeah. Thank you.
So thank you, Shannon, once more.
Thank you.
And I would like to thank all the speakers of this morning, and you can run for coffee now. And you should be back 20 to 11, I guess. So about this time, Gatra again here. Thank you.
Hello, hello. Hello. Hello, hello. Yes, good. So, welcome back. Excuse me. So, welcome back to this session, which is the, I think, I don't want to say, is the last session of the very last day of IFRO. I think from my experience, we have had an amazing journey for last 3 days or more, 4 actually. So first I wanted to thank everyone who are here still with us, and also those who are online. I think we have more than 21 participants online. Actually, they are also the key, and we have in this session 3 remote speakers. We are going to listen to them. And before I introduce our next speaker, I want to say that I'm Amit, and I am going to moderate this session for you. And we have 2 speakers, first from Czech University of Life Sciences, who are our colleagues. So I'm very proud to introduce them. So without wasting much time, I want to introduce our first speaker, David Mussolek, who is going to speak about the feeding and growth of 2 invasive defoliating insect species on 3 native European elm species under 2 irrigation regime. And David, the stage is yours.
Hello, nice to see you all here, despite it's Friday and almost the end of the conference. And also thanks to the organizers to make this nice conference. And I would like to introduce you a study which we did here in the Czech University of Life Sciences, Prague, where we deal with the native European elms and some invasive insect pests and the interactions with under some different regimes of water stress. As you probably know, the most common European elms are the 3 species: Ulmus glabra, Ulmus levis, and Ulmus minor. And since the last century, there is a decline of those species caused mainly by some biotic agents like the Dutch elm disease caused by the ophiostoma. And the elms also suffer by some other biotic or even abiotic stresses. And additional to those are some new alien species which may cause some damage to the elms. One of these is the elm zigzag sawfly, Aproceros leucopoda, which you can see on the upper left picture. There is a female laying an egg to the on the leaf. And then the larvae hatch, and then they make such a typical feeding trail in the zigzag pattern. Well, let's say here is a nice example of how it looks like. Oh, sorry, this is an ad, but we can skip it. Yeah, the larva is feeding this way. The larvae go through usually through 5 larval instars. Yeah, then they pupate. So, wonderful, isn't it? Yeah. Okay. And this species occurs in Europe since 2008 or so. And then it spread from Eastern Europe to the Western part. And now it's also in America and other countries. And it originates in Asia, in Eastern Asia. Okay, and it creates occasional outbreaks when the trees can be also almost defoliated. And the most important thing is that the species is specific to elms, to the Ulmus genus. And the other defoliator which we deal with is an aphid. It's also from Eastern Asia, Tinocalis takashihoensis, and it also invaded Europe. There are more countries where this species occurs, and it also creates some occasional damage to the trees, which you can see, for example, on the left picture. And yeah, and to determine to evaluate the insect performance on the trees, we designed an experiment using the 3 European native Ulmus species, Ulmus levis, glabra, and minor. And we had 40 individuals of each species, and half of them were irrigated ad libitum by water, and the other half suffered by permanent drought. And we tried to introduce the insects to the trees in this design, where we introduced only the aphid Tinocalis to the trees, or only the larvae of first instar larvae of the Aproceros leucopoda, or both of them. And we also had some control trees without insects. This was repeated 3 times during one vegetation season. It happened in 2020. This is how it looked like. To keep the insects on some special place, we installed some nylon sacks on the twigs with several leaves, and into the nylon sacks we introduced the insects, and we kept them there for about 2 weeks. And at the end of each of this repetition, we measured either the development success, the size or the weight of the pupa of Aproceros, number of damaged leaves, number of attempts to feed, and the leaf area which was consumed by the larvae. And in the case of the aphid, we measured the population density, or the amount of individuals per leaf area after the time period. And then we calculated also the reproductive rate. And for the whole trees, we also evaluated the overall vitality, the occurrence of the aphid, because it was virtually impossible to keep them only inside the nylon sacks. They also, part of them also invaded to the whole trees. And we sampled also the leaves for biochemical analysis. It was before introducing the insects and at the end of the insects feeding. Yeah, so here are our results, some of the main results. Those are related to the feeding or the performance of the Aproceros leucopoda. The main obvious thing was that this species was not able to finish its development on the Ulmus levis at all. The larvae tried to eat the leaf, but then they died, all of them. In Ulmus glabra, there was really very high mortality, but few larvae were able to finish their development. and only the ulmus minor was well accepted by the Aproceros leucopoda. This is very similar with the amount of feeding attempts. As I said, for ulmus levis, always at least one feeding attempt was present, but not many more. In ulmus glabra, there were few feeding attempts, and ulmus minor was damaged the most. There was no significant effect of the irrigation treatment. In terms of time, there was some decline of the leaf area consumed at the second half, or in the summer, let's say, when the larvae consumed less amount of leaf. It also, something interesting is that there is an interaction with the aphid on the leaves, where there were the Aproceros together with Tinocalis, with the aphid. Then the Aproceros was smaller, yeah, and it even declined in later summer. So there is some kind of competition, probably. Yes, those are results related to the Tinocalis takashioensis. There is also some preference, but not for ulmus minor. It is for ulmus levis. It was the most preferred species by the aphids, but they were also very well able to live on other ulmus species. And similarly, there is some negative interaction, like competition with the Aproceros leucopoda. So in the nylon sacks, where there were together the Aproceros with Tinocalis, there was some lower density of Tinocalis after the observation period. Yeah. And in terms of season, there was also some decline of the Tinocalis density in the later times of summer. This is the reproductive rate, which means more than one means that the population is growing. The population of Tinocalis, less than one means that it is declining. So there is some variability in the season, but those are just some preliminary results. We will dig more in the data and try to find some other patterns or so. And most importantly, we will try to combine this data with results of biochemical analysis, which will be presented by the next speaker. Yeah, and this is all from me. And thank you for your attention. And I'm looking forward for any questions. Thank you.
Thank you, David. So we have, I think, you're well in time. So we have a scope for a few questions from the audience, as well as I think we can see also our online attendees if they have any questions. So far, no. Anybody? I think people are thinking, you know. So in the meantime, I think I should ask one simple question, like as I'm working on bark beetle, and we always say that drought stress induce more attack in the trees, right? And in your result, you showed many times that drought has no impact on. How do you justify that the drought is not doing? Because sometimes we see in the, considering the plant physiology, that if you have a drought, then they have the choice. One, either they invest energy to defense or growth, you know. So there is a situation the plant has to choose, you know. So and it happens sometimes in drought. We saw different papers published and showed this kind of dilemma in the plant. So in your case, I think we see there is no impact of drought. How do you justify that?
Yeah, thank you for the question. Actually, from the results which you saw, there were no very significant differences. But as I said, those are just some preliminary results. Maybe at the end we will find some differences among the irrigation treatments. There were some differences in the biochemical analysis, which, however, you will see in the next presentation. But it was also our hypothesis, or we expected there will be some differences in the insect performance in between the two irrigation treatments. Maybe the stress was not enough, let's say, but as I remember, the trees were really heavily stressed, as the leaves were time to time also wilting, and so at the moment I don't have really good idea how to justify those results, but maybe in combination with the results of the biochemical analysis, we will see more.
Okay, thank you, David, for your answer. I wish you good luck with your experiments. And again, thank you. So we should say, so it's my pleasure to introduce our next speaker, Dr. Elena Kaliyukova. She is also working in our faculty, and she will be talking about, I think, the chemistry part of the earlier story. Her talk of the title of the talk is Study of Phytochemical Changes in Ulmius species infested by invasive sawfly and aphid under different level of water stress. The floor is yours.
Thank you, Amit, for introduction, and my many thanks to organizer of conference and for the opportunity to present our study. And I'm analytical chemist, and I would like to show you what happens with your samples when they get to the laboratory. And our study is, my presentation will be connected with previous David study and analysis of 3 almost species. So first of all, it's very important to prepare samples before biochemical analysis, and very important to, in the right way, collect the samples. After the samples, require some samples pretreatment before extraction of bioactive compounds, following with different instrumental analytical method for analysis, and after that we are able to do some statistical evaluation of the results. Almost samples were collected during the season once a month in June, July, and August. This sample leaves were directly freezed in liquid nitrogen. Totally, we collected 560 samples and analyzed them. When the samples were freezed, they should be lyophilized in order to save all the bioactive compounds in the leaves. And after that, the samples were gomingized using oscillating mill. For the analysis, we selected 2 instrumental methods. It's LC-MS cutoff analysis of carbohydrates. This method was established in our laboratory, and we are able to separate such carbohydrates like sucrose, fructose, glucose, raffinose, mannitol, myonisitol, arabinose, and tregalose in one run. And also we selected well-known spectrophotometric methods for determination total phenolic, flavonoid, non-tannins, and condensed tannins content in order to see general trends and changes in the samples. For determination of carbohydrates in UMU samples, extraction procedure consists of two parts. In the first part, we took only a small amount of the samples, approximately 20-40 milligrams of the samples, and extracted it in a mixture of methanol, chloroform, and water. After that, the mixture was hidden in the water for 30 minutes, and after that, this extracts should be purified. In this purpose, we used Dobbeck's cation and anion resins. And only after this step, we are able to analyze the samples using the LC-MS Q2 system. For spectrophotometric analysis, another procedure was applied. We took 50 milligrams of the samples, extracted it firstly using a detail editor in order to remove some pigments and fats from the samples. This procedure was repeated 3 times to be sure to remove all of these contaminants. And after that, we extracted the samples using acetone. As well, we repeated this procedure 3 times, and after the extracts were combined and used for analysis of total phenolic contents and total non-tannins content, we used following Chikatilo method. For determination total phenolic content, we use aluminum fluoride colorimetric assay, and for determination total condensed tannins, we used butanol assay. Now I would like to present some of the results that we found that, of course, during the season, some changes are visible in phenolic, condensed tannins, and flavonoid contents. But also we found that, for example, in Ulmus glabra, the concentration of these compounds is lower in comparison with Ulmus laevis and Ulmus minor. As well, in carbohydrates analysis, we found that under drought stress, the concentration of fructose is high in comparison with well-irrigated samples. As well, that in Ulmus glabra samples, the concentration of this sugar was lower in comparison with laevis and minor samples. Glucose concentration was the trend was similar like in analysis of fructose samples. The concentration in Ulmus glabra was lower in comparison with samples of Ulmus laevis and Ulmus minor. But also we found that the sucrose concentration doesn't have a strong influence, and it didn't change in different stressed plant samples. So the drought stress and water stress didn't have a strong influence on sucrose concentration. And the next carbohydrates that we determined in the samples was myoinositol. This carbohydrate served as osmoprotectant, and we found that at the beginning of the season its concentration was lower, but with in a period of time the concentration increased in 2 times in all samples. So the next step in our research will be metabolomic analysis, because we found that these samples are different, and we would like to identify compounds which influence on the test, which influence on nutritional quality of the tested ELMS samples for the selected insects. And in summarization, you can see on the slide. And of course, my many thanks to our colleagues from EVA project and of course to Axel Schopp from the University of Bochum. And as well, thank you for your attention.
Thank you, Elena. So we have scope for questions. Anybody? I think I see that you explained the methodology, like material method part is very nicely, and some of the students are taking quick notes. So that is good. I appreciate that. Normally in our presentations, we don't get much time to detail the material method part. But I just wanted to have one simple question that you did this three, if I understood correctly, you did the 3 plants, you did this assay, LC-TOF, and you did, and you have also drought stressed plants, right? So do you think all these 3 plants responds to drought stress similarly, or is there any pattern, or they are different in their response to drought stress?
Thank you for your question. And I think that we will find some similar compounds which would respond on drought stress. But I'm not sure that these 3 species will have the similar trends in these compounds. So we have to check and to continue our study.
Okay, thank you. I think the metabolomic study will be very interesting to see what is actually happening there. It's more detailed way. So once again, thank you to Elena for her nice talk. So now it's time to move to our remote speaker. So let me introduce to our first remote speaker from Canada. So welcome, Ilga Port, and she will be talking about next-gen sequencing based biosurveillance for Asian Longhorn Beetle. Quite interesting. So the stage is yours.
Thank you very much for introducing me. I just want to let you know that we had some problems with the bandwidth, so I switched off my video just for the sake of understanding. Anyway, I switch it off again during the question period. Can you see my screen? Yes.
No.
Okay. Can you see my screen? Can you see my screen? Yeah. Okay, thank you, because I couldn't hear you.
Just wait a minute. We have some tech issues here. Just give us 1 minute.
No problem.
I cannot hear her. Yeah, you may start now.
Okay, thank you very much. Anyway, good morning. Good morning again. Thank you very much to Sandy and the rest of the organizers for giving me the opportunity.
Ilga, please give us time. I think we are not.
Okay.
Ilga, can you please check your microphone, because we cannot hear you.
Okay, can you hear me? Can you hear me?
We can hear you in our headphones, but not in the mic, in the audio. So just give us a few more minutes.
Sure, no problem. No problem.
YouTube is kicking us out. And can you try again, please?
Hello?
Yes, yes, perfect.
Okay, great. Anyway, so thanks very much for giving me the opportunity to talk to you today while remotely. This is unfortunately beyond my my influence, that it had to be for me online. Well, thanks again. And now I would like to talk about our work that we are doing in a Genome Canada funded project, which is called Biosafe. Biosafe stands for Biosurveillance of alien invasive species. And bio or genomic-based biosurveillance is of a pest is proposed here to facilitate early intervention relating to prevention and preparedness, which is deemed less costly than later interventions, where costs dramatically increase with past time of inactivity. And so my talk is about genomic biosurveillance resources that we developed for the infamous Asian Longhorn beetle, abbreviated ALB, which is an important quarantine pest in Europe, the United States, and in Canada. So now I have to continue. Okay, next slide. So my talk is divided in 4 sections. I will address briefly the problem. Maybe most of you are already aware of this pest actually, and then proposing very high level a preparedness solution. Second, I will be talking about our results from population genomic studies of the ALB in its native environment, and also talk about contemporary gene flow, which we found in its native range and the extent of this gene flow. And third, I will also report on sort of early success in ALB invasive pathway reconstruction for biosurveillance purposes. And lastly, fourth, I will talk about how can we operationally apply this gained knowledge for rapid and accurate identification of this serious pest to help regulatory authorities. The Asian Longhorn beetle, so as I mentioned, it's abbreviated often as ALB, mostly feeds on poplar, willow, elm, and maple in its native China, while it's also, it occurs also in Korea, but mainly in China. I will put the focus on China today. So while in the invaded areas, the ALB preferentially feeds on maple trees, and this of course poses a serious threat, especially to the multi-billion-dollar maple syrup industry in Eastern North America, should this pest become established. Usually ALB has 1 generation per year, and ALB's lifestyle is depicted on the right. I have no time to go through it, but so I just want to mention that adults tend to stay and reinfest the natal host actually, and they rarely fly. They have very low flight capacity. They move unless the host quality declines or they are disturbed by a predator. There are important telltales of ALB infestation on trees, which are the oviposition pits, the frass, the large exit holes, and also twigs with stripped bark. And one of the most important reasons actually for ALB's success as an invasive pest is its physiological adaptation to cold. ALB larvae are known to be highly freeze tolerant thanks to their increased production of low molecular weight sugar alcohols such as glycerol, during which they accumulate during chilling. Just briefly to mention that the ALB genome has been sequenced and was published in 2016 in Genome Biology. So this will actually help us a lot in our endeavor to actually map reads on a reference genome. It is still very fragmented, but still good enough to help us in SNP calling. So the draft genome is below 1 gigabase, and there are around 22,000 genes in that ALP genome. And one of the focuses of the annotation of this genome was actually genes that make this pest so successful as a wood-boring pest. So genes that basically are capable of degrading wood are highly diverse and expanded gene families. As I mentioned already, so one of the things that make ALP so successful is really the cold hardiness of its larvae. With regards to the worldwide distribution, ALP is native to China and Korea. So it first drew attention in the 1980s in China. And then it was also introduced into North America with infested wood packaging material. It was detected also in Europe, like in Austria in 2001, but also in France and Germany. And as of July 2021, there are 33 countries that have reported ALP occurrences, and several eradication efforts have taken place in the invaded areas, but ALP can still be found in North America and Europe, and these beetles are also frequently intercepted. So there is a lot of uncertainty actually, and we heard about that already during this conference, about an invasive's origin and likely pathways of introduction. And this fact complicates actually decision-making regarding mitigation and management. So therefore, within our project Biosafe, we collected global populations of top priority forest invasive species, 4 in total, and among them was the Asian Longhorn beetle, where we sequenced of these menaces, their genomes, or produced reduced representation sequencing information, like in the case of the Asian Longhorn beetle, and also determined their biogeography in the native range. And we also conducted functional genomics experiments to better understand the pest's capacity to invade successfully, for example, by studying the genomic determinants of cold hardiness in ALP, which was done at a different university in our project. So our project already identified genetic markers that are useful for the development of genomic biosurveillance tools based on target enrichment. So this is our final genomics tool, where we have already preliminary information about the loci that are of interest to us, in order to identify species, assign source populations, and also predict risks of spread. So to develop said genomic-based biosurveillance tool for the Asian Longhorn beetle, firstly, we had to do vast sampling with a lot of collaborators, and we covered most of ALP's native Chinese and also invaded North American areas. And then we conducted reduced representation genome sequencing and genotyping, also known as genotyping by sequencing, to identify single nucleotide genetic variants along the genome to examine the genetic population structure through model-based or non-model-based clustering methods, and reconstruct the ALP invasion history. For the native ALP populations, we performed sampling in China and Korea, as you can see on the map on the left. And so we sampled 20 distinct sites. In China, our sampling covered major geographical regions, as you see here color-coded, namely the Northeast, the North and Northwest, and also South populations. Our results showed that the Korean ALP samples were genetically distinct from the Chinese ALP samples. And this is true for the Korean ALPs that we actually sampled in forested areas. So these are more than the natural habitats of these beetles. However, for a specimen that we obtained from urban areas, from seaports, it was different. We found actually that those beetles nested within Chinese ALP population. So this is also shown actually on the right side in this maximum likelihood tree. So we see some differences between these different habitats. So more natural versus more urban, more So within China, we found by doing spatial principal component analysis that there is a major north-south break, which divides the populations into two genetically distinct groups. So, and this is actually corresponding what we see also with other insects and even plants in China. We did also discriminant analysis of principal components, and we identified 6 genetically distinct clusters among the ALB populations, thus exposing additional genetic substructure within each of the 2 major groups. And we also assessed contemporary gene flow using a software called BIAS-S. Overall, we found low levels of gene flow among all populations. But we did find gene flow, and not only within 2 northwest populations, but also gene flow from the northwest into the northern China plain. And we also identified some, while minor, gene flow among northeast populations. And our results indicate, while large-scale contemporary human facilitated migration seems largely absent, directional gene exchange within and between certain regions, and here I want to mention the northwest and the north, do exist. So in order to reconstruct the invasive pathways for samples that were intercepted in North America, we sampled in 5 US states, but also in Ontario, so specifically in Toronto, Canada. So again, we jointly called SNPs together with the native ALB samples, and we obtained a global set of samples and SNPs of almost 600 samples and 500 samples. After filtering, it came down to 500 samples and around 3,600 SNPs that we could use. So again, we use this reduced representation technology, which is called GBS, genotyping by sequencing. And the first striking results was that the heterozygosity for the invasive populations was low, and compared to the US samples, the Toronto, so the Canadian samples showed even lower heterozygosity overall. So which could suggest there have been some sort of genetic bottleneck events at play, which resulted in a reduction of genetic variation. In the principal component analysis, we found that the first principal component was able to differentiate several populations, including Toronto, Ohio, Massachusetts, and also New York Farmingdale. And we also investigated whether 2 distinct infestations in Toronto, so in red, you have the circles and the triangles, are clustering together, as this would mean that the ALBs from the second infestation are likely survivors of the eradication attempt of the first infestation. So we could actually confirm, yes, it was the case. These were actually, we assumed that these were survivors actually of the first eradication attempt. And in the maximum likelihood tree, we also found Toronto ALBs that were distinct, but they were nested within a clade that regroups native North Chinese ALBs. For the purpose of population assignment, we performed population clustering of native ALBs, which is depicted on the left as the smaller dots. And then we used these population clusters to predict the source populations for the invasive individuals, which is depicted as the solid squares. And we used DAPC again now for population assignment. So in this so-called contingency table, which shows the actual clusters for the invasive ALB populations in the columns, and then the inferred clusters related to the native ALB populations are shown in rows. And from these results, we can easily see that we proposed there were 2 major sources of invasive Asian longhorn beetle populations. So that, and this is from the North China plain, and it's depicted here in blue, which is CHE, which is Chengde from Hebei province. And the second one is GI, so Jinjiang from Shandong province, into Illinois, so Chicago, New York, New Jersey, as well as Ohio and Toronto, respectively. Of note is also the assignment of the 2 invasive populations found in Massachusetts. You can see this here, also the second and the third row. To the Korean population ULS, which is Ulsan. But our study, as I mentioned previously, has already revealed that this Korean population is genetically similar to a Chinese ALB population from the southern part of the North Plain. So basically, that this Chinese ALB population is actually the actual source population. So based on the discriminant analysis, and also we used another software that we developed in our project, which is called Microbiome, which is based on machine learning and also phylogenetics. We came up with a set of 500 discriminant SNPs from the global population analysis, which includes the native as well as the invasive populations. And we designed something called ion ampli-seq panel, and for the LB we called it Beetles. So the basis of the genotype analysis, of this type of genotype analysis, is multiplex PCR and the ion torrent sequencing technology. So meanwhile, the panel was validated using phylogenetic analysis to reproduce a population structure using the selected loci. And we recently received the data from 500 to 600 samples acquired through our collaborators for our first ampli-seq panel test run. And we will further proceed with validation and blind testing steps of the ampli-seq tool to move towards the final operational tool for intercepted samples. So to summarize, our genome-wide analysis was able to uncover decent population structure in native ALB with limited gene flow between regions. These results are a prerequisite to use genetic markers successfully in population assignments and in developing the genomics-based biosurveillance tool. And that could be of great help to regulatory authorities such as the Canadian Food Inspection Agency, which is our main project partner. And I didn't have time to talk about it, but we did also, meanwhile, a genotype environment analysis to come up with some candidate genes for cold hardiness in the populations. And we identified some promising candidates, and that will be cross-validated with a transcriptomic study on larval fault tolerance that is being performed at our partner, Western University. And then last, I want to thank, first of all, for you to listen to my talk, but I also want to acknowledge several other people behind this work briefly. Firstly, my PhD student, Ming Kui, who was the part of her PhD thesis I presented. On the right, you have Isabelle Giguère, who is the overall project and lab manager, Resham La, the project lead of Biosafe, Amanda Rowe. She's very much involved in the ALB activity in the Biosafe project. And both are also Ming Ming's thesis co-supervisors. And I also want to acknowledge our collaborators from Europe, China, who provided us with the new samples, and also the US, who helped us with putting together an impressive ALB population sampling. And last but not least, I acknowledge our funding partners of the Biosafe project. And if you want to have more information about other aspects of this project, please visit the website, which is updated regularly. And thank you very much, and I'm happy to take questions. I hope I didn't get too much over time.
Thank you so much. Thank you. So we have some time for questions. Anybody?
Yeah, I was the director of a thesis of Marion Javal, who provide you the samples. And so my question is simple. I noticed that you two have no samples in the southern and southwestern part of China, where Anoplophora is present and even abundant. So do you think that if you integrate new samples from southwestern China, Yunnan especially, and Sichuan, it will change the whole issue of genetic analysis, especially, for instance, the northern plain. We also know that they correspond to plantation. So it's probably introduce ALB from other parts of China. So not the native area of China, of ALB.
Yes, so we said, so the south, which is not the south, right? Oops. Hello?
Yeah, we can hear you now.
Hello?
Yeah, we can hear you now.
Okay.
Hello.
Yeah, sorry, I was just kicked out of the internet. Yes, so our sampling is more, I would call it geographically more east than south. In the south, there is actually not really Asian longhorn beetle. I was told it is actually the citrus longhorn beetle who looks similar, but it's not the same. And they don't even, or, well, we tested some hybrids, but suspected hybrids, but they were actually Asian longhorn beetle. We did have samples, so I didn't present that, but our collaborator Yongki Wu from USDA, he did some sampling in the southwest. So we have some additional populations, and he did a microsatellite analysis on them. So there is, so for example, we could see a little bit more movement also out of the southwest. This is true. So that, but overall, actually, so we actually that data that he gave us, or that we also presented in our paper that we just submitted, actually confirmed our population structure. So we did not find any very much new results here. So it was, but we did find a little bit of a movement out of that area as well.
Thank you. I think we are running out of time. So I think we need to stop this question and answer session now. So thank you again for your nice presentation. So let's move on to our next remote presentation. We'd like to welcome Nero Lilly from Kenya Forest Research Institute, and he will be talking about the invasion pathways and effect of Scrum antisecum on forest landscape ecosystem in Kenya. So the floor is yours. Hello. We cannot hear you.
Hello.
Yeah, it's fine. Yeah. Please carry on.
Okay. Thank you. My name is Nero Lilly, and thank you for the opportunity to make this presentation. I work for Kenya Forest Research Institute, and I'm going to talk about invasion pathways and effects of Cestrum auroticum on forest landscapes ecosystem in Kenya. A case study of Western Mau Forest block. Mau Forest is a critical ecosystem for the country because it's the largest water catchment in Kenya. This report was prepared by a team composed of experts from forest, socioeconomics, governance, and livelihoods, and ecologists in areas of invasive species and ecological modeling. Yes, as a background, this species of Cestrum belongs to the Solanaceae family, and it's native to North and South America. It's a shrub which is evergreen, making it very suitable for ornamental purposes. However, in Kenya, this species is very invasive, especially on the montane forest, the high altitude areas, because of several characteristics it possesses, such as the rapid growth, as well as it matures very fast. At the age of 2 years, this species starts to flower and it produces a lot of seeds. Moreover, this species can be propagated by several mechanisms, including seeds and vegetative waste. The disadvantage of this species is that it colonizes the foresty floors very fast, and it suppresses the native regenerates, which is a great threat to biodiversity in the long term. These are some of the photos showing a heavy undergrowth of cestrum in major plantation species in Kenya. So we undertook a study with 3 main objectives. One was to understand how these species got introduced into the study area, as well as the spatial distribution at the moment, the perceived socioeconomic effects of the invasion, and the effect of the cestrum invasion on ecological processes, especially the forest regeneration. To understand the spatial distribution, we undertook a mapping where we established line transits that ran from the edge of the forest towards the interior, and it traversed several landscape types, such as the native forest, the plantation forest, the roads, among other landscapes. And we established plots systematically at an interval of about 500 meters. And we used these plots to assess the population structure of cestrum, as well as the ground cover of cestrum. And we subjected these maps to QGIS to generate the distribution maps. To understand the perceived socioeconomic effects, we undertook household surveys where we use semi-structured household questionnaires, and the information was triangulated using key informant interviews and focus group discussions. The responses were subjected to weighted scores, and data presented in terms of and descriptive analysis was undertaken. On the effect of invasion on forest regeneration, the plots which were used for mapping, small subplots were used to enumerate the regenerates, and the data was used to determine the species richness of the regenerates, the diversity index, mainly the Shannon diversity index, and density of the regenerates across different classes of cestrum cover. And this data was collected last month during the wet season. Yes, from the findings, it's clear that a number of challenges is facing the Mau forest ecosystem. And notably, the 2 greatest challenges is forest grazing and invasive plant species colonization. However, these 2 challenges usually affect the understory vegetation, meaning that this forest ecosystem is on the verge of loss in the near future. Yes, in terms of introduction and spread, this species of cestrum was introduced in Kenya intentionally in 1921 for ornamental purposes. However, it found its way into the study area way back in 1970. And through the process of reproduction and dispersal by birds, this species has been able to spread into the natural and plantation forest in the study area. And it was only until 2013 when there was an alarm of the extensive spread of this species in the forest ecosystem. Currently, the species continues to colonize the ecosystem. In terms of the perceived trends in the population buildup of this species, a majority of the respondents opined that this species had increased in population in the last 5 years, and in the next 5 years, it's likely also to increase. And they attributed the potential of the increase in population due to the rapid means of dispersal of this species. This species is largely dispersed by birds, as well as it's also heavily propagated through the vegetative means. And also the species grows rapidly, meaning that it has a high potential of expanding its range very fast. For the positive and negative effects associated with this species, yes, majority of the community associated the species with the death of livestock. When livestock consumes the fruit of this species, deaths are reported within 24 hours of consumption. And the other challenges, which are very notably, is the displacement of grazing lands and the native species. Some of the positive effects of this species is these species actually do not have important economic uses, apart from being used as an ornamental plant and for fuel wood. And for this fuel wood, it's only used as a lighter to light other fuel wood, because it cannot be used on its own, because its wood has a very choky smoke. And looking at the absolute scores of the positive and negative effects, it clearly indicates that the positive effect of this species cannot compensate the negative effects it has. In terms of estimated economic losses, which is majorly loss of livestock by the local communities, a household can lose up to $270 on annual basis. And because the community acknowledges the negative effect or the threat of this species, most community members are willing to contribute to the management of this species by contributing about 15 USD to support the control of these species in the ecosystem. Looking at the spread of this species in the Mau forest, yes, the white dots in the map represent the areas which species was not found. The red dots shows the areas where the species was spotted, and the red big ring is where the species was first introduced in this study area. And clearly, the species was spotted in about 75% of the mapped areas, meaning that it has extensively covered the ecosystem. And in terms of occurrence, this species is non-selective on the landscape, as it was found across different landscapes, including the plantation forest, the natural forest, and the farmlands. In terms of dominance on the foresty floor, about 30% of the foresty floor is covered by this species. This indicates that this species is still building up its population. And if eradication measures are put in place at the moment, there is a possibility of eradicating the species. But as the population builds up, it would not be possible to get rid of this species in the near future. In terms of population structure, majority of the cestrum shrubs were in mature and seedling stages. But I would like to draw your attention to the differences in population structure between the dense forest and the open forest. Ideally, it is expected that open forest would contain more cestrum seedlings than dense forest, because it's well known that open canopies encourages regeneration. But this was contrary, as shown in this chart. And the possible reason is that at first, this open forest encouraged rapid colonization of the species. But however, as the regenerates recruited into mature shrubs, the mature shrubs usually occur in clustered clumps, which are very thick. And the possible reason for the low regeneration of the new seedlings is the rapid suppression by the mature cestrum shrubs. But for the dense forest, although there's a rapid colonization of cestrum, a few of them are able to recruit to mature trees because of the shading effect of the tree canopies. Yes, because this study found that there is a negative correlation between the canopy of the trees as well as the ground cover of cestrum. As the canopy of the native trees increases, the ground cover of the cestrum declines, meaning that this species is most likely sun-loving and it cannot thrive well in closed canopies. Yeah, these are photos showing extensive growths of this cestrum in open grounds in natural forests. In terms of effects on forest regeneration, the study clearly found that invasion of cestrum tend to reduce the species richness and diversity as well as density. As the ground cover covered by cestrum increases, the richness of the regenerates decline from 22 to about 3 species only, while the diversity declined from about 1 to about 0.5. Similarly, the regenerates, the density of the regenerates significantly declined as the ground cover of cestrum increases. These are photos showing a thick or heavy regeneration of cestrum in natural forest. In effort to control the spread of this species, CAFRI has been undertaking a number of capacity building activities to build the local community on how to control its spread through uprooting these plants at young stage of establishment before they start flowering, and rehabilitating those areas immediately with fast growing tree species so that it can suppress the emerging seedlings. In conclusion, it's very clear that this species is widespread across the landscape, and it poses a number of negative socioeconomic and ecological effects because it clearly it colonizes the foresty floor very fast, displacing the native species, which reduces biodiversity and ecosystem functionality, as well as affecting the livelihoods and well-being of the community members. In way forward, there is an urgent need to control the spread of this species, as well as protect the areas which have not been invaded from invasion. Because as it continues now to take over from native species, it means this forest ecosystem are under threat of existence in the near future. Therefore, there's also need for extensive awareness creation, as well as coming up with more technologies on how to prevent the spread, as well as eradicate these species. And I wish to take this opportunity to call for joint efforts and partnership on coming up with physical ways on how to control these species. I would like to acknowledge CAFRI for funding this study and Kenya Forest Service for collaborating in this study, as well as the Community Forest Association and community members, and all those who assisted us in collecting this data. And I say thank you, and I welcome questions.
Thank you. We might have one question. Yes, please.
Hi, this is René Eschen from CABI. Thank you for the very comprehensive and clear presentation. I was surprised about the difference between the economic impact that people perceive as a result of especially loss of livestock, I think, and the relatively small amount of money that they're willing to spend on controlling of the species. Can you explain that difference, please?
Hello, can you hear the question? Nero?
Yes, yes, please. Yeah.
Did you hear the question?
Yes, I got the question. Maybe I could ask Abdallah to respond. Abdallah, please. Abdallah or Dr. Langas, please.
Thank you so much, Nero, for the opportunity. From the figures that we see, the economic impact is we're going to focus on the mode. Of course, it's very high considering the economic status of these people. But if we had produced more results, we would have seen that the community perceived the government as the person responsible to be able to take this, the management of this species, and that's the small willingness to pay figure that we get. Thank you.
Thank you to both of you, Abdullah and Nero. And now, actually, so let's move on our last speaker of the session and last speaker of this IFRO symposium. So we are pleased to welcome Victoria Lansner, and she will be speaking about drivers of global scolytiny invasion patterns. So the stage is yours, Victoria.
Okay, so can you see the screen?
Yes.
Okay, well, hi everybody. I'm very glad to be here presenting this talk. I will start now. So I guess you may already know very well that the international movement of goods and people associated to the globalization process has historically caused accidental introduction of species outside of their native range. And many of these species have important impact following their establishment. So consequently, there's a need to understand the global drivers of invasions. And if we see the geographic variation in numbers of established non-native species around the world, we note that this may reflect both historically variation in the propagal pressure as well as the variation in habitat characteristics of the new region, right? The invasibility of the new region. So analyzing the relative importance of these factors can help developing biosecurity policies aimed at minimizing the numbers of future invasions. So in this study, we focused on the scolytine subfamily. As you may know, this comprises a very diverse group of insects with about 6,000 species. Several species in this group are considered important agents of mortality in forests around the world. And additionally, these species have a great invasive potential for being easily transported in raw logs or wood packaging material used in international commerce. So consequently, this group represents a very interesting case study for understanding global patterns of invasion. So the aim of this study was to analyze global patterns of historically scolytine invasions and to explore the effects of factors that may explain these patterns. So we compiled all the available records of scolytine species established outside of their native range. And for each species, we compiled information on both the native and the invaded range, classified into 6 biogeographical regions, the ones I'm showing here. And we also classified each species based on their feeding habits in bark beetles, ambrosia beetles, and seed or twig feeders, and based on their mating system in breeders, so species for which ship mating has been documented, or outbreeders, species for which mating only occurs between unrelated individuals. And on the other hand, we identified a set of variables characterizing each region that could potentially explain the observed patterns of invasion. So these were the cumulative imports. We compiled information on the value of imports between each pair of regions from the period between 1870 to 2014 as a proxy of propagal pressure. We also estimated the availability of habitat in each region by estimating the land cover by forest in each region. We estimated a climatic similarity index between each pair of regions. And we also obtain information on the number of native Scolytine species in each biogeographical region as a proxy of potential competition with invasive species in the native in the new regions. So this is some of the results we found. Here I show the geographical patterns of exotic species in each region, and we found records for a total of 123 species established outside of their native range. In blue, I show the number of native species to each region that established in other regions, and in red, the number of exotic species established in each region. So the main pattern here is that the Palearctic and the Indo-Malayan region, so these two regions, have the highest total number and proportion of native species that invaded other regions. On the other hand, the Arctic and the Neotropic regions are the ones that have the higher number of non-native established Scolytine species. So if we say if we see the relationship between pairs of regions regarding the number of exotic species, we found that by far the highest fraction of species from a region that invaded other region was from the Palearctic to the Nearctic, and then followed by species from the Indo-Malayan region to the Nearctic, and from the Palearctic region to the Neotropic. When we analyze the volume of imports, the highest trade volume between regions, also by far, occurred between the Nearctic and the Palearctic in both directions, and followed by imports from the Indo-Malayan to the Palearctic, or Palearctic to the Indo-Malayan regions. And then regarding to the climatic similarity between regions, we found that the Nearctic and the Palearctic were more similar with each other than with any other regions, while the Neotropic, the Indo-Malayan and the Afrotropical regions, the areas with forests of these regions were very similar with each other, while the Austro-Pacific region was was not similar to any other region. So when we perform a multiple regression model to analyze the relative importance of all these variables in explaining the percentage of species native to a region that invaded other regions, we found that for the three feeding habit groups, the volume of imports was the most important variable explaining the invasions, and it was in all cases, it was positively related with the percentage of invading species. And additionally, for seed and twig feeders, there was the number of native species in the destination was also important in explaining the number of invasions, and it was positively related. So this suggests that there is no negative effect of competition with the native species or a biotic resistance at this large scale. We also analyzed the total number of regions colonized by each species, including their native region. And as expected, we found that most of the species established in two regions, so their native regions and one other region, and only a small fraction, these are 10 species, established in all the 6 regions. So when considering the feeding habit, we found that most of the bark beetles established only in a few regions, in 2 or 3, and only a few fraction of these species were able to establish in 4 or 5 and none in 6, while ambrosia beetles showed a larger proportion of species able to establish in a large number of regions, and for seed and twig species, almost the same number of species were able to establish in 2 or 3 regions or in more than 3 regions. And when we analyze the same species, but looking at the mating system, we found that for outbreeding species, most of the species also established in a low number of regions, in 2 or 3 regions, while inbreeding or species able to inbreed or seed mating were able to establish in a large number of regions. So it should be noted that most ambrosia and seed-feeding species are inbreeders and bark beetles are outbreeders. So there's a relationship between these groups. So to conclude, the numbers of non-native scolytin varied considerably among the 6 pore regions. The Nearctic and the Neotropic are the most invaded regions, but have not been source of large number of species invading elsewhere, while on the other hand, the Palearctic and the Indo-Malayan regions are the main sources of exotic species, but a relatively low number of exotic species have established in these regions. We also found that ambrosia beetles and seed and twig feeders, for which inbreeding is a common mating system, are more successful in invading a larger number of regions. This is probably because inbreeders might have a lower allele effect threshold that outbreeders, since mating takes place among siblings before dispersal, and this allows avoiding the difficulties in mate location. And last, the value of cumulative import was the strongest variable in explaining the global scolytin invasion. And well, this likely reflects that wood packaging material used in international commerce provide an excellent pathway for this species. And in this sense, it's important to notice that change in trade policies between regions in the future may involve new sources of invasions of scolytin species, and consequently it highlights the importance of intensifying biosecurity programs and phytosanitary treatment of imported commodity, as well as border or post-border surveillance in order to minimize future impacts of these invasions, particularly in previously isolated regions. So that's all. Thank you very much. And if you are interested in reading more about this study, you can find it published.
Thank you, Victoria, for your nice and informative talk. So do we have any comments and questions for Victoria? Yes.
Thank you. This is Jean-Claude Grégoire from Brussels. I was interested in this study because I participated in another study similar within the framework of EPO, and beside that we are finishing a third study. What strikes me is the general nature of your conclusions. I mean, relative to more surveillance at the borders and relative to some types of pathways, such as wooden packaging material, and the more particular nature of your study, where you tried to identify particular drivers of invasion. And to me, it would appear that at the end, we are not going to be able to use the particular driver because there are so many exceptions, and that finally we might have to turn into more general regulations regarding the pathways instead of identify particular features in particular bark beetle groups. What is your feeling about that?
Yeah, I guess, I mean, my results or my study was at a very large scale, and there was no detail in the particular pathways. So I think that the interesting thing here is to try to see the big picture and see the different international or routes that of commerce that there can be, and to be aware of these risks. But yes, I don't know if we can give much detail on what to look at with these big patterns. It's just to try to understand what happened in the past, and why we are seeing the results of past commerce or trade politics, and probably with the change in the treatment of materials, of packaging materials, these patterns will change, and we will see it in the future. So we can't predict much about what will happen in the future, I guess.
Okay, Victoria, thank you. I think we are almost 15 minutes out of our schedule. So thank you again, Victoria. So before we approach our closing ceremony, just we all have to say goodbye to our online participants. So thank you all for being with us, and stay tuned. Thank you. So now